behind the peritoneal cavity, and there's so much back that to see the kidneys, you pretty much have to remove all these other organs, all of them. So you remove, for example, this liver here, abdominal cavity. You remove the stomach, and you remove the large intestine and small intestine here. You just remove this large intestine piece here and pancreas. And there you go. You see these kidneys here? They're way back, right? Way back. And you can see the aorta, inferior vena cava. So from the aorta goes red color renal artery, and blue color is renal vein. And you see the ureter, the tube going down, ureter. And this ureter opens into this thing, the bladder. And bladder goes out to the urethra. In the male, urethra is longer because of penis. In female, urethra is shorter. So these are the parts here. And if you look at, let me see. Same thing is shown here also. Again, you see the kidneys, right? You see renal artery and the vein, inferior vena cava and the aorta. And what is this tube here? Ureter. And you can see this going to the bladder, right? Bladder. This is a male with a prostate. This is a female with the two ovaries here, right? Over here. And in the female, you also have the uterus and the bladder. And in the male, you have the prostate and other structure that I will tell you in that system. So what are these two things here? Ureter. Also what happens is, it is showing ureter opening here. We should be showing lower down here. And what happens is that you have two openings of the ureters, right? The bladder, and one opening going out, correct? Urethra. So three openings. So that three opening area is called as triangle. Okay. This is a triangle. And when we study the bladder, then it will be mentioned here in the bladder. Today we did the kidney, it's not mentioned here. Okay. And if you look at the kidney structure, you should come closer here. You can see from here. So you have this part here is the cortex, and of course there's a capsule here, right? And this is the medullary pyramids, and this part is the pelvis. Between the pyramids, what is this area called between the pyramids? Renal column. Between pyramids, renal column. This thing here, this part here is minor calyx. And minor calyx is the joint to make what? Major calyx. And major calyx joint to make the pelvis. Pelvis goes into the ureter. Alright? Let's talk about the blood vessels. One more time. You can see your handout. See the renal artery, segmental. Then this is lobar here. What do you call this between the pyramids? Interlobar. And from interlobar going like an arc like this. Arcuate. See, it's like an arc between medulla and the cortex, right? And then from this arcuate artery going like this in the cortex is interlobular. From the interlobular, now look here. This is the line here. This is the cortex. This is arcuate artery, arcuate vein. From arcuate artery going like this is interlobular. From interlobular going to glomerulus. See this ball-like thing here, red? Is glomerulus. Going to the glomerulus is what? Afferent with an A, right? Afferent arteriole, not artery, arteriole. And it doesn't show the other one. So this is the vein, the arcuate vein, and so on. So this part is cortex, this is capsule. And below this line is the medulla. Alright? So, in this nephron, this is one nephron and the nephron. Which one is this? What kind of nephron? This versus this. This is cortical, is more towards the cortex, smaller loop of Henle. The one which is more in the medulla is called a juxta medullary nephron. But anyway, both have the same parts. This part here, you can call it as Bowman's capsule. It's better called Bowman's capsule because it doesn't show. See, this is the detail here. Here, this is Bowman's cup. And you see the glomerulus here, right? Bunch of capillaries, glomerulus. And you see here the bigger one, the smaller one. How to identify whether it is afferent or efferent? Afferent is bigger, efferent, exit is narrow. So this is bigger one, so it must be afferent with an A arteriole. The narrow one, smaller one is the efferent. So that creates a pressure in the glomerulus because exit is narrow, so you're basically forcing. You force, and it gets filtered here. You remember openings are there in the capillaries, many openings are there. Through that blood gets filtered. Okay. Outside these glomerulus, outside this, you notice these cells here? What are these cells here? Podocytes. These podocytes have filtration slits, they help with filtration. Podocytes. And this space here is called capsular space. So when the filtrate comes out, it comes into this space. And this wall here is peripheral layer of Bowman's capsule. So this whole structure, the entire structure is actually renal corpuscle. In this whole structure, renal corpuscle, there are two parts. One, glomerulus, which is a bunch of capillaries. Two, the cup-like thing, right, surrounding it. The cup-like thing has inner layer is this foot cells here, inner layer. Outer layer is this one here, the parietal layer. And what is this space here? Capsular space. What are these cells called? Podocytes. What is this here? Afferent arteriole, and this one? Efferent arteriole. This one here, don't worry, okay? Just ignore for now. This one here. So, here, going back here, this one here is this, right? You can call it as Bowman's capsule, or you can call it renal corpuscle, you may call it glomerulus, whatever, right? It's not showing all the details here. So you can call it Bowman's capsule. And then, from here starts renal tubule. Renal tubule has how many parts? Three parts. And those three parts are, the first one, start from here, this one here. What is this? The first one is proximal, right? Proximal convoluted tubule. And follow this, it goes down, right? Going down, descending limb into the medulla. Going back up into the cortex, ascending limb of what? Loop of Henle. And then it coils again. What do you call that? Distal convoluted tubule. Then opens into this duct here. What is this duct called? Collecting duct. So to know whether it's proximal or distal, to know whether it's descending limb or ascending limb, you have to start from glomerulus, right? Start from here. Because first come PCT, then descending limb, then ascending limb, then DCT. Any questions here? Okay. What else we have in your handout for the lab? Collecting duct is the big tube here, this big tube here. So basically urine that has been made by the renal tubule will go into this collecting duct. So collecting ducts are here. They go like this. From collecting ducts, it will pass on to minor calyx. From minor to major calyx, pelvis, ureter, and so on. Okay, what else do you have? Histology, okay. Let me show you the slide. I think you don't have dissection. Let me see here, if you have kidney here. If you have kidney here, let me show you how it looks like from inside. Okay. Let me show you.
Isn't it wonderful?
You can see this in the animal kidney. You can see the ureter, right? Ureter now. You can see this side is hilus. And to see the capsule, this is membrane capsule here, see? Right, capsule. And if I cut this capsule, is the capsule here, right? Okay. And to see the structures inside, you can give it a cut in the center or maybe off center, whichever way you want to give. Let me give a cut slightly off center, longitudinal cut. See this inside here? You can tell here that this is the cortex here, right? And this part here is the medulla. And this thing here does not show minor major calyx because it is actually inside like this. Let me remove this to show you minor calyx. Let me remove this. You see this minor calyx? Right? You see minor calyx here? I just removed some of these tissue here. So if you remove this, let us see. Okay, so here is the minor calyx here, and major calyx and pelvis. So this is what you need to identify from this. You don't have to dissect it, just one cut. Sometimes we get colored. That's much better because what they do is they drain out all the blood from the animal, and then they inject latex, colored latex, in the blood vessel, the artery red, in the veins blue, so you can see. So sometimes it is tricolored, bicolor, tricolored, so then you can see the different structures here, but not in this one. Okay, so you don't have to dissect this. So next thing I will show you is histology.